Hi and welcome to EaseGenetics. I'm Dr. Kaveh Zargeri, and in this video, Genetics Terminology Part 1, we'll start by breaking down basic concepts like alleles, genes, and chromosomes in simple, relatable terms because understanding these terms gives you more confidence understanding basic genetics. My main goal is to make it as easy as possible. Let's start with chromatin. Chromatin is a complex of DNA and proteins found in the nucleus of eukaryotic cells. Genetic material is located inside the nucleus like an intertwined bundle or like a can of worms. You can't say if it's a one piece or of several pieces. It is a compact structure, but still the necessary genes that a cell needs are accessible. It's not very dense and compact, yet not as open as possible. This is the state that genetic material is functional and the cells use the genes for their needs. Chromosomes are thread-like structures composed of DNA and proteins, mostly histone types, found in the nucleus of eukaryotic cells and in a similar form in prokaryotes. In eukaryotes, chromosomes are paired, called homologous chromosomes, having been inherited from the parents, one from each. This term is usually used for simplicity, but this state of chromosomes can only be observed during a brief cell cycle period, and the genes are not functional. I'll get back to this. At the two ends of each linear chromosome, there is a region called the telomere. It is the protective end region consisting of repetitive nucleotide sequences and associated proteins. Since chromosomes have adhesive properties, the telomeric regions at the ends of chromosomes act as a kind of a sealing mechanism to prevent them from sticking to one another because the two ends of DNA are kind of sticky. Each chromosome has usually one region called the centromere, which is a specialized region of a chromosome that plays a critical role during cell division. It is the point where the two sister chromatids of a replicated chromosome, which we call it sister chromatids, are held together, and it serves as the attachment site for spindle fibers via a protein complex called the kinetochore. There's no genes in centromeric region. It divides a chromosome into two arms. The upper arm is called the P arm, showing with a small p, and the lower arm is called the Q arm, with a small q. Chromosomes can be seen in four forms based on the location of their centromere. Starting from left to right, when the centromere is in the middle of the chromosome and divided into two equal arms, we call it a metacentric chromosome. Then we have the submetacentric chromosomes. The centromere divides the chromosome into two unequal measurable arms so we can measure each arm. Next is the acrocentric chromosomes, which the centromere is located near one end of the chromosome, but yet a small arm can be seen, but we can't measure its length. And finally, the telocentric chromosomes, which the centromere is completely at one end of the chromosome, so these chromosomes have only one arm. This characteristic of chromosomes is one of the methods to identify homologous chromosomes. Autosomal chromosomes are the non-sex chromosomes found in the cells of an organism. In the animal kingdom and some plants and, of course, humans, except for one and some rare cases that have a few pairs of chromosomes which are sex chromosomes, the rest are called autosomal chromosomes. For example, in humans, there are 22 pairs of autosomal chromosomes labeled 1 through 22 based on their size, with 1 being the largest and 22 the smallest. These chromosomes are the same in both males and females and carry the majority of an individual's genetic material. A karyotype is a visual representation of an organism's complete set of chromosomes, organized and displayed in a standard format. It is typically used to study the number, size, shape, and structure of chromosomes in a cell. In a karyotype, we have to arrange the chromosomes in pairs according to their size, centromere position, and banding patterns, which are made visible using specific staining techniques like Q banding, R banding, etc. The chromosomes are usually in descending order or of size, with the sex chromosomes X and Y placed at the end. So the largest chromosome will be chromosome number 1, the next tallest will be number 2, and so on. And remember, in a karyotype, the microscopic image of the chromosomes is used. Sister chromatids are two identical copies of a single replicated chromosome, connected by the centromere. Before the cell division starts, either it's mitosis or meiosis. In the S phase, the DNA, which is in the chromatin state, replicates, and before the cell division starts, the DNA amount of the cell becomes double, and in the late prophase stage of mitosis or prophase 1 of meiosis, as the chromatin becomes compact, we can see it as a sister chromatids. Now here is what I want you to see in one vision. Chromatin, sister chromatids, and chromosomes are all the same thing, genetic material. The genetic material simply changes shape at different times and stages, taking on a specific form in each phase. During most of the cell cycle, it exists as chromatin, being active and functional. Before cell division begins, in the S phase, the genetic material replicates. By the end of prophase or prophase 1 and in the metaphase stage of cell division, it is seen as sister chromatids, and after that we see such things as chromosomes. Now, in the comments, tell me which stage you can say you are observing chromosomes. So now let's get deeper in the genetic material. A gene is a fundamental unit of heredity in living organisms, composed of DNA or RNA in some viruses. Genes carry the instructions for building, maintaining, and regulating the structure and functions of cells. A gene is essentially a part of a DNA molecule that is functional, and it expresses a specific form of a trait. For example, it might result in blue eye color, red petals in a flower, or coat color in an animal. A locus, which its plural is loci, is a specific fixed position on a chromosome where a particular gene or genetic marker is located. The term is used in genetics to describe the physical location of a gene or sequence of DNA within the genome. For example, on human chromosome number 15, there are two genes responsible for eye color. This means that these genes are located in a specific region of chromosome 15 in all of us. But there is a term called translocation that can change the location of a gene, which is usually a rare phenomenon and discussed in cytogenetics. An allele is a variant or different forms of a gene. Different alleles arise due to variation in the DNA sequence of a gene, mostly caused by mutations, and they can result in different traits or characteristics being expressed in an organism. In fact, if genes had only one form, there would be no genetic variation in traits. A gene must have at least two forms or alleles, or more, such as flower color in a plant for two types of red and white, or three like the human blood groups, which have three alleles, A, B, and O. Some traits can have more, even up to 10 alleles. There are interactions between the alleles of a gene. In one case, one allele is dominant and the other is recessive. A dominant allele is an allele that expresses its effect on the phenotype even if only one copy is present. So one dominant allele is enough to see its expression. A recessive allele is an allele whose effect on the phenotype is only observed when two copies are present. The dominance of one allele over another allele could also be shown as big A is larger than small a, as you see on the right. In genetics, a trait is an organism's specific characteristic or feature that we can see or measure. Traits can be influenced by genetic information inherited from the organism's parents, environmental factors, or a combination of both. They can range from physical attributes such as eye color, height, and hair texture to biochemical and physiological characteristics like blood type or the ability to metabolize certain substances such as insulin or enzymes. Phenotype is the observable physical, biochemical, and behavioral characteristics of an organism, which results from the interaction of its genetic makeup with the environment. Examples of phenotypic traits include height, eye color, blood type, and resistance to certain diseases. So when you're talking about someone's height, your friend's eye color, or the color of a flower, you're talking about their phenotype. Here's an important point. In genetics, we often start with the phenotype of an organism to get to its genotype. So let's see what is genotype. Genotype is the complete set of an organism's genetic material, specifically referring to the specific alleles or genetic makeup for a particular gene or set of genes. It serves as the blueprint for the development and functioning of the organism, encoding the information needed to build and regulate cells, tissues, and organs. In simple terms, the genotype is an individual's genetic makeup. It represents the alleles that the individual has inherited from their parents. By naming the alleles, the genotype of an individual consists of the two alleles they have inherited from their parents. For example, if a gene has two alleles, big F and small f, the genotype of an individual could be one of the three possible combinations listed on the right. Now, when we look at an individual genotype, it could be homozygous, which means the individual has two identical alleles for a particular gene or genetic locus. These alleles may both be dominant, big A, as you see on the left, or both recessive, small a, on the right. Or could be heterozygous, in which the individual has two different alleles for a particular gene or genetic locus. In the middle, you see a heterozygous individual with big A and small a. I hope these were useful to you and made a better understanding of these terms. Thanks for watching this video. I hope it was useful to you. If you enjoyed watching this video, please press the like button. If you want to watch more videos, please press the subscribe button. Feel free to share your thoughts, questions, or suggestions in the comments below. I hope to see you in the next video. Bye.